In this class, we'll be dealing about taxonomy. We'll be looking at phylum, the first classification, first chapter for zoology that deals with more of protozoa, porifera, hemichordata, and onychophora. And before that, we'll just look at what taxonomy basically is, how we classify the animal kingdom in short, and the general classes that are in place or the general division plans that are in place in taxonomy. We'll look at acoelomate and coelomate and pseudocoelomate animals. We'll look at symmetry as well, protostomes and deuterostomes. I'll be covering questions related to this chapter for zoology for past 10 years. So let's have a look at it. So the term taxonomy was coined by Candle, D. Candle. Taxonomy is a science of classification of organisms as we are aware of. Two important criteria based on which taxonomy is or animals are divided or categorized or taxonomized is one is the evolutionary relationship of animals and one is the shared features among the organisms. Now, let us have a look at the general classification plan that is widely accepted. So the basic or the shortest category is species. Species, genus, family, order, class, phylum, and kingdom. This is the general 7 kingdom or 7 plant classification that we follow. Now, under the species, there are certain infra kingdoms that we have or small kingdoms. These will be subspecies, clades, and deans. Now, based on the classification plan, organisms can be categorized into major and minor phyla. How are organisms categorized into this? So, based on two important factors is what the classification is done. When we take a phyla, the number of species in that phyla will determine whether the phyla is a major or a minor phyla. The second important character is the relation or the participation of the species or organism in the ecological communities. So when we look at factor 1 and try categorizing organisms, we will see 11 major phyla. But then when we look at factor 2, we'll see that 2 of the phyla belonging to the invertebrate categories will not come under the major categories. One is the Ectopropta phyla and one is the Rotifera phyla. However, the most amazing part to see here is that these two phyla comprise about 80% of the animal kingdom or invertebrate kingdom. But then again, their role or these organisms do not interact in a major factor as part of ecological communities. Hence, these have to be brought down to a level of minor phyla and we will not be dealing about them in detail. Now, let us have a look at the kingdom Animalia, how we categorize this kingdom into the subsections. This is the most important part that we will be dealing with. Eumetazoans in detail, protostomia and deuterostomia, and this is an important part about which questions have been asked in the past 10 years. So, main questions that we could see in the IAS Zoology paper 2 or paper 1 for mains were coming from this part, parazoa. One is deuterostomia and protostomia. One is symmetry, radiata and bilateria. How do you categorize radiata and bilateria? What is the normal symmetry that comes in? So I'll have questions for you, or I have prepared the questions from previous years that you can have a look at. I'll show you the questions. Let's have a look at the question. This is what we will be covering with in detail today. So what is meant by symmetry? This has been asked multiple times. Write down different types of symmetry in animals. What are the minor phyla and list minor phyla with examples. So what I told you, Ectopracta and the other one. So we'll have to deal about that. But then again, this is not something that we'll be dealing in today's class. I'll take a separate session for you guys regarding this. Okay. So define coelom. How coelom is developed. What is a coelom? What are the different types of coelomate animals that we see? So differentiate between major and minor phyla again. Describe salient feature of any one phyla. Then what is bilateria? Explain the regions or theories of origin of bilateria. Protostomes and deuterostomes. Straightforward questions from this chapter, right? Yeah. So kingdom Animalia. So kingdom Animalia, it's a huge kingdom. So basically it's divided into two sub-kingdoms. One is protozoa and one is eumetazoa. Now what protozoa deals is, it deals with unicellular animals. This eumetazoa will deal with multicellular animals. Now eumetazoa, for the sake of understanding, is categorized into 3 infra kingdoms. One is mesozoa, one is parazoa, and one is enterozoa. Now mesozoa and parazoa is a little bit confusing because both of them will have porifera in it. But porifera has 2 categories again. Some of the porifera are going to be primitive type of porifera, one are more advanced type of porifera. So primitive type of porifera, we will be classifying it as mesozoa, and the more advanced ones we will be looking at it under parazoa. Now enterozoa, again for the sake of classification or for simplification, has been categorized into two. One is radiata and bilateria. So this is again based on symmetry. Symmetry plays an important role because see, based on the symmetry, the symmetry has been developed in organisms based on their dieting habits or locomotion types. So when you are considering symmetry, I'll tell you what the different types of symmetry are. We'll have radial symmetry, we'll have radial, spherical, and biradial symmetry. So biradial symmetry now comes in organisms wherein the organism is more motile for its feeding habits. But radial symmetry, etc., comes in an organism which is stationary or sessile, where the food is abundant in its atmosphere or surrounding medium. So we'll look at it. So then bilateral, because it is more common, or in more advanced species we see bilateral symmetry, we'll classify it into deuterostomes and protostomes. This is again based on the development of the blastopore. What the blastopore in an organism becomes. Generally, when we see development, the blastopore in an organism develops into an oral opening in protostomia, but it develops into an anal opening in deuterostomia. So based on the variation in that or development of the blastopore, we'll have a look at them in detail. Now protostomia will have three categories under it: acoelomate, pseudocoelomate, and schizocoelomata, and deuterostomia will have enterocoelomata. Now these two, schizocoelomate and enterocoelomate, are called eucoelomates or true coelomate animals because both of them have true coeloms in them. But difference comes in based on the development of coelom in them. Now schizo, this word, schizo means it's derived by cutting the mesoderm. So schizo in Greek or Latin means cut. So schizo means a cut in the mesoderm. So the mesoderm that is there while the organism is developing in the embryonic layers, a cut or indentation takes place in that, and a coelom is formed, which is why it is called schizocoelom. Whereas in enterocoelomata, what happens is that the mesoderm forms pouches, and these pouches, or you might have heard about archentron. Archentron, are you familiar with archentron? No, so I'll have to tell you in that detail. So what happens is that when we are developing from an embryo, we'll have the blastopore and the archentron. So the archentron is the basic entity from which all the germ layers come into existence. So we will have the ectoderm, endoderm, mesoderm coming in place. So the archentron has an inward involution, or it convolutes internally. So if you are taking into picture the archentron in the shape of a jug, the archentron will fold on itself. It will fold towards inside, and this internal fold takes form of a coelom. So this is going to be called as enterocoelomata. Whereas in this one, this jug-like structure of the archentron closes, and there is an indentation that comes on, just like a cut. It comes as a cut, and this cut forms the coelom, which is called a schizocoelomata. It's clear, right? If you want, I can explain. It's clear? Alright, so this in detail. See, protozoa, unicellular animals, one phylum included, that is protozoa. Eumetazoa is metazoa, multicellular animals with well-defined tissues. Subkingdoms include mesozoa, parazoa, and enterozoa. Now, mesozoa is infrakingdoms includes animals from porifera. Parazoa, this also, as I told you, includes porifera but with specialization, advanced porifera. Enterozoa, again into two grades, radiata and bilateria. So, as I said, radiata is diploblastic, two layers only, ecto and endo only, and triplo is, it has a mesoderm in between. So, this will mostly have a radial symmetry and diploblastic body. Schneideri is an example, and this is mostly advanced eumetazoans, a triploblastic body. Okay. So we'll talk about cleavage mechanism in these two when we deal with in detail, because with radiata, what we observe is that it shows mesozoic cleavage. So you guys are aware of the symmetrical planes, right? Transverse, longitudinal, and oblique planes, right? Great. So again is acoelomata, pseudocoelomata, and schizocoelomata. So they have a true coelom, which is formed by splitting of the mesoderm, lined by peritoneal layer, phylum Annelida, arthropoda, mollusca are included. And deuterostomia, as I said, anal opening is formed from the blastopore, mouth is formed later from the blastopore, the opposite end. In deuterostomes, cleavage pattern is radial and indeterminate. You understand what is indeterminate? Okay, so with the cells that are formed from the embryonic layers, right? In animals like the primitive animals or animals that show or animals that come under protostonia, what happens is that these cells, once they multiply, they lose their ability to multiply again or lose their ability to form an entire organism again. So that is form of a determinate division. But we advanced chordates and human beings have indeterminate development, which means we have the potential, our cells have the potential to bring forward a new being or a new organism if the need requires. That means the cells can develop into any new type of cell. So that is known as indeterminate type of development. So enterocelomata is only one. So enterocelomata, പറ. ഡിറ്റർമിനേറ്റ് എന്ന് പറഞ്ഞു കഴിഞ്ഞാൽ നമ്മളെ ബോഡിയിലുള്ള സെൽസ്, നമ്മൾ ഒരു സെല്ല് നമ്മളെ ബോഡിയിലുണ്ടെങ്കിൽ ഒരു പ്രിമിറ്റീവ് ഓർഗാനിസം ആണെങ്കിൽ അതിന് ഡിവിഷൻ ചെയ്ത് കഴിഞ്ഞാൽ അതിൻ്റെ പിന്നെ മൾട്ടിപ്ലൈ ചെയ്യാനുള്ള ശേഷി പോകും യൂഷ്വലി. അതാണ് പ്രിമിറ്റീവ് ഓർഗാനിസത്തിലും ലോവർ ഓർഗാനിസത്തിലും കാണാം. എൻലിഡ്സ് വരെ അതാണ് ഫീച്ചർ. വി ഡു നോട്ട് സീ ദം മൾട്ടിപ്ലൈങ് അഗെയിൻ. ആ സെല്ലിന് ആ ശേഷി അങ്ങ് പോകും. പക്ഷേ അഡ്വാൻസ്ഡ് ഓർഗാനിസത്തിൽ ആൻഡ് ആർത്രോപോഡയ്ക്ക് ശേഷം നമ്മൾ കാണുന്ന ഓർഗാനിസത്തിൽ നമുക്ക് കാണാൻ പറ്റും സെൽസ് ഡിവൈഡ് ചെയ്ത് കഴിഞ്ഞാലും ആ സെൽസിന് ആ പൊട്ടൻസി ഉണ്ട്, ടോട്ടി പൊട്ടൻസി എന്ന് പറയുന്നില്ലേ, മൾട്ടിപ്ലൈ ചെയ്യാനുള്ള ശേഷിയുണ്ട്. അവർ പക്ഷേ അത് എപ്പോഴും ചെയ്യണമെന്നില്ല. ബിക്കോസ് ഹ്യൂമൻസിൻ്റെ കേസ് എടുത്താൽ മെയിൽസും ഫീമെയിൽസും കൂടി ഒരു ഇൻട്രാക്ഷൻ ഉണ്ടായി കഴിഞ്ഞാൽ അല്ലെങ്കിൽ സെക്ഷൽ ഇൻട്രാക്ഷൻ ഉണ്ടായാൽ അതൊരു ഡെവലപ്മെൻറ്റിലേക്ക് പോകും. പക്ഷേ വി ഹാവ് ദി പൊട്ടൻഷ്യൽ. നമ്മുടെ സെൽസിന് ഇപ്പം ഫോർ എക്സാമ്പിൾ നമുക്കിങ്ങനെ പറയാം, എൻ്റെ കൈ മുറിഞ്ഞു എനിക്കൊരു വൂണ്ട് വന്നു, അവിടെ എപ്പിത്തീലിയം പോയി, ആ എപ്പിത്തീലിയത്തിന് പിന്നെ ഡെവലപ്പ് ചെയ്യണം. പക്ഷേ എൻ്റെ ആ വൂണ്ട് ഉണ്ടായ സ്ഥലത്ത് കണക്റ്റീവ് ടിഷ്യൂ മാത്രമേ ഉള്ളൂ. But the situation requires my connective tissue to proliferate. ആ ഒരു ശേഷിയാണ്, my connective tissue can proliferate upon reaction and it can cause or it can form an epithelial lining there as and when it is called about. This feature is called indeterminate development. അപ്പോൾ ആ ഇൻഡിറ്റർമിനേറ്റ് ശേഷി നമ്മൾ അഡ്വാൻസ് കോർഡേറ്റ്സിലും ആനിമൽസിനും ഉണ്ട്. അതാണ് അവിടെ. Now, coeloms and coelomates again. ഇത് നിങ്ങൾക്ക് ഞാൻ സ്ലൈഡ് തരാം. So this will, I'll give you the slides. So this is all you need to write. But then again, zoology, when you take it, it's a straightforward subject, okay? But then more than you write, you will have to draw. The way you fetch marks in zoology for mains is by drawing. As much as you can draw, the better. And one more tip that I can give you is that this is a huge subject. It's a vast subject. You will have to, animal kingdom itself, I'll have to take it maybe 13, 14 classes. It's a huge class. So I found it hard when I did my medical. So when we study or compare organisms, it's pretty hard to remember all of it. The best way to do is draw diagrams for each class. Keep it as short diagrams, right? You can draw and keep it in 1 page or 2 pages and keep on referring to it. Based on the diagrams, remember what the anatomy is. What is the circulation type? What is the respiratory organ involved? What is the type of nervous tissue involved? If you remember all this or learn how to draw this or represent an organism, it's the best way to learn. Because theory മകപ്പ് ചെയ്ത് പഠിക്കാൻ പറ്റില്ല. ബയോളജിയിൽ നമ്മൾ എല്ലാവരും ചെയ്യുന്നതാണ്, പക്ഷേ ബയോളജി നടക്കില്ല. So it's the easiest way to do that. So coelomates and acoelomates. So a coelom, as I said, is a hollow cavity, is fluid-filled cavity between the alimentary canal and the body wall, lined on all sides by mesoderm. What we consider as a true coelom. Let's discuss that. So if you take a jug and keep a jug inside that, the space between the two jugs is what we'll be calling a coelom or a coelomic pouch. But is the space sufficient to call it a true coelom? No. We will require the space to be lined by mesodermal cells on both sides. In some of the slides you will see peritoneal cells being written. So from the peritoneum is what the mesodermal cells come in. So what I mean by peritoneal cells is mesodermal cells. Don't get them. Okay, ഞാൻ പറഞ്ഞു, നമ്മളിപ്പോൾ ഒരു ജഗിൻ്റെ അകത്ത് വേറൊരു ജഗ് വയ്ക്കുന്നു. അപ്പോൾ അതിൻ്റെ ഈ രണ്ട് ജഗിൻ്റെ ഇടയ്ക്ക് ഒരു സ്പേസ് ഉണ്ട്. ആ സ്പേസിനാണ് നമ്മൾ സീലോം എന്ന് പറയുന്നത്. ആ സ്പേസ് ഉണ്ടായാൽ മാത്രം പോരാ, ആ സ്പേസ് കാണിക്കുന്ന പ്രിമിറ്റീവ് ഓർഗാനിസമും ഉണ്ട്. അപ്പോൾ അതിനെ നമ്മൾ സീലോം എന്ന് വിളിക്കില്ല. ആ സ്പേസിൻ്റെ രണ്ട് സൈഡും ഫോം ചെയ്യുന്ന ജഗില്ലേ, ആ രണ്ട് സൈഡിലും മീസോഡോമൽ സെൽസിൻ്റെ ലൈനിങ് വേണം. എന്നാൽ മാത്രമേ അതിനെ ട്രൂ സീലോം എന്ന് വിളിക്കുക. അതിനെ പറഞ്ഞുതരാം. നോക്കുക, acoelomate, in which no coelomic cavity exists. Examples: flatworms, coelenterate, sponges. Only a gut, coelentron or sponges seal exists in these animals, and there is no cavity. അവർക്ക് ഒരു സിമ്പിൾ ഒരു വാ മാത്രമേ ഉള്ളൂ. സ്പോഞ്ചസിൻ്റെ നമ്മൾ ഫീഡിങ് ഹാബിറ്റ്സ് കണ്ടു കഴിഞ്ഞാൽ ദേ only have a gut. അതൊരു മോഡിഫിക്കേഷൻ ആണ് സ്പോഞ്ചസിൻ്റെ. They were sessile organisms. They used to have lot of food But then again, once their dieting patterns changed, they found that it's easy to stay at one place because they have a lot of food surrounding them and swallow. So to save energy, they lost their ability to move around. They became sedentary, and they developed the cells that were lining the outside of the organism, sponges, the cilia that were lining sponges, they migrated towards inside of the animal, and they formed a sac-like structure, which is called the gut. And what used to happen was that the cilia used to move, and all the water used to enter the organism's gut, and they used to digest the organic material, and they used to throw out the waste through the same opening. Sponges have only one opening. അതാണ് പോറിഫറയുടെ ഏറ്റവും വലിയ ഒരു അഡ്വാൻറ്റേജ്. ഒരു ഫൈലം പോറിഫർ, ഇഫ് യു ലുക്ക് റൈറ്റ്, അതൊരു പ്രിമിറ്റീവ് ഓർഗാനിസം. ഇറ്റ്സ് നോട്ട് ഇവോൾവ് ഫോർ മോർ ദൻ 570 million years. ഫസ്റ്റ് ഇവോൾവ് ആയ ഓർഗാനിസം ഒരു ഗ്രൂപ്പിൽ നിന്ന് ഒന്നാണ്. ആൻഡ് പാലിയോസോയിക് ഇറയ്ക്ക് ശേഷം അത് ഡെവലപ്പ് ചെയ്തിട്ടേ ഇല്ല, അങ്ങനെ തന്നെ നിൽക്കുന്ന ഒരു ഓർഗാനിസമാണ്. സോ ദാറ്റ് ഈസ് വൺ തിങ്. സോ ഈ സീലോമേറ്റ് മനസ്സിലായല്ലോ. ദർ ഇസ് നോ സീലോ, ദർ ഇസ് ഓൺലി വൺ ഗട്ട്, സീലൻട്രോൺ, സ്പോഞ്ചോസീൽ, എക്സിസ്.